I'm curious about why certain animals in nature can regenerate limbs, tails, all kinds of different structures, and what sets them apart from humans as animals that can't regenerate very well. My name is Josh Currie. I'm an assistant professor of biology at Wake Forest University in Winston-Salem, North Carolina.
You've done research on zebrafish and basically other organisms, should I say?
So I was part of a team, each lab as part of that, a different organism. And we specifically looked at different vertebrates, so animals that are like us that have backbones. And we kind of used three different organisms that have kind of different body plans and different regenerative potentials. So zebrafish specifically, are a really powerful research organism and they can regenerate lots of different structures, but they don't have limbs. But they do have fins that regenerate, and so those are kind of an evolutionary precursor of what limbs are like. And then what our lab studies is the salamander, the axolotl, and these are kind of the only limbed animals, so animals that have limbs that look a lot like ours that can fully regenerate those limbs anywhere that they're amputated along their axis. And then David Brown's team at Duke looked at mice, and mice as mammals like us, they can't regenerate limbs, but like humans and mice, they can actually regenerate the very tips of their fingers. And so they use that as kind of a proxy for regeneration in axolotl limbs and zebrafish fins. And so we were able to compare these three different species and try to find commonalities that might exist across their regenerative potentials, even though they're very different.
And so, was there something that came out of this research that how mice or mammals could regenerate limbs?
One of the take home messages is that there are some commonalities between even animals that are distanced over evolutionary time by millions and millions of years. When they do undergo regeneration, they do have some commonalities. And specifically, what we were thinking about was the skin that covers that regenerating structure. And we know from fish and from salamanders and even some data from mice, that the skin that covers the regenerating part is very special. It's not just a normal kind of skin that covers an injury, but it actually has some specialized roles. And and what we found was that there are some common signatures, some common genes that are shared between fish, axolotls, salamanders, and mice. And so that was actually really encouraging that there are some common pathways that each of these very different species are using in order to make regeneration work.
What's next in your research, and what are you looking to do next? What's the next step along this path?
For us specifically, we're hoping to kind of keep our this comparative work going. But one of the things that we're really interested in is that we actually use CRISPR genetic scissors to get rid of one of these commonly shared genes. And what we were really excited about is that it seemed to have a really crucial role in the axolotl's limb regeneration. And what we saw is that when we, basically cut out this gene from the axolotl genome, the animals tried to regenerate their limbs, but they didn't really get it right. They were losing parts of their skeleton as part of the regeneration process, so the robustness and the fidelity of that process actually went down really, really dramatically. And so what we're excited to do is maybe dig into that and actually try to understand what is actually happening on a molecular level, what's happening on a genomic level that this gene, which is called SPA, is doing kind of during regeneration.
I mean, I guess that there are other genes as well that are in the whole process that help and identifying what those are in order to possibly, like, theoretically add them into a human genome?
Yeah, exactly. So SPA is what's called a transcription factor. It's living inside the nucleus of a cell and it's helping turn on or turn off different genes inside the nucleus. And so there are probably hundreds and hundreds of genes that are missing when SP8 is gone. So what we're really trying to identify is what are some of the crucial things that it's controlling and how does that impact the entire process. And Yeah, and obviously, as I mentioned, the skin that is over this regenerating structure is really unique, and so in other contexts where regeneration doesn't happen, we know that special regenerative skin doesn't apply. And so one part of the work actually was trying to actually use like a gene therapy to see if we could substitute for all the things that potentially this special regenerative skin is doing. And so we were partially successful in that. And I think it was kind of early days, but the idea is that potentially in places where regeneration doesn't happen, where it doesn't create this specialized skin, that we might be able to substitute in different molecular factors that can help kind of either spur kind of regenerative skin to form or potentially kind of substitute for some of its functions.
The process of regenerating, let's say, an arm seems so complicated. It seems like it's almost impossible. Do you feel like that could be a possibility in the future for humans to regenerate their arm or their leg?
It is a daunting task to think about regenerating an entire limb. And I do think that there are lots of people that are in regenerative medicine or in regenerative biology that are taking many different approaches. That actually gives me hope because I do think my personal opinion is that it will be kind of a multidisciplinary approach that will need people that are, of course, studying it in animals that are able to do this. And maybe we can identify some clues, but there are also folks that are working from kind of an engineering standpoint, seeing if they can build kind of bioreactors that can grow tissue in a lab, building scaffolds, unique kind of prosthetics, and also folks that are in synthetic biology trying to kind of in stem cell biology, thinking about how we can create stem cells that can recreate. So my personal opinion is that it's really going to be kind of a it's going to take all of these folks to work together in order to come up with a solution because it's daunting when you think about, a salamander limb is only maybe a couple millimeters or a couple centimeters, but a human limb is obviously much, much bigger. So being able to recreate a patterned amount of tissue that is as big as an adult limb is going to be a really challenging problem.
Yeah, because I mean, normally it can take someone 18 years, right, to grow that limb fully.
Even for salamanders, it takes time, and we even see that from, the small animals that we use for experiments to a large adult animal, like timing does scale. So I think that's why there is gonna have to be rather than a shot that you receive, if you happen to lose a limb. I think it will be kind of it will require biologists, engineers, stem cell biologists all working together to create something that can accelerate the time frame.
What about regeneration not only of limbs, like regeneration of, spinal cord, or I don't know, even maybe cartilage or things like that? Are you thinking about those things as well?
We've kind of specialized in the limb, but of course, the axolotl is able to regenerate a lot of different structures, and it's pretty astounding. And I think one of the things that we're really thinking about is really what diverts healing towards something that is like regeneration versus just kind of normal wound repair. So when that limb is amputated, why does it actually create this proliferative mass of cells that we call the blastema? Why does it divert towards regeneration rather than just kind of healing over and that being the end of the story? And there are lots of examples like that in regeneration where there should be just a normal resolution of injury, but for some reason, the tissue kind of goes further and starts to recreate the structures that are missing. So, for instance, in the spinal cord, the animal, if it loses part of its tail, the regrowth of that tail actually includes the spinal cord. So, it can kind of create de novo a new spinal cord off the distal end. But there are examples if there is a crush injury, would mimic most humans experience, leading to kind of a loss of leading to paralysis in the axolotl, that injury is resolved, but not only that, but it actually recreates the spinal cord in the place where there was that crush injury. So it's able to not only resolve the injury, but then kind of take that next step to kind of reconstitute all the missing structures that were lost. So I think that's really the unique thing that we're thinking about is why does it take that next step to actually reconstitute the missing structures rather than just kind of, make sure that the injury is resolved.
Maybe there are certain parts of the body of the axolotl that it just heals it, and other areas that it does regenerate, right?
Yeah, I think that's a really good point that and even what we see in mammals and other animals, there are certain locations that are primed or able to regenerate and those that don't. So for instance, even in the mouse, we know that the very tip of that finger can regenerate, but if there's an amputation that's just a little bit further back, it can't regenerate. So just that simple location can make a big difference. And if you think about across the animal kingdom, if you think about reptiles, so things like lizards, they can kind of regrow some proxy of their tail. If you've ever seen like a skink or something lose its tail, it can regrow kind of an approximation of that structure, but lizards are not able to regenerate their limbs. There is something about certain structures that kind of maintain some kind of competence to regenerate. And so that's also something that we're thinking about in the lab is how do structures not only how do they regenerate, but how do they maintain the ability to regenerate? There must be some kind of readiness factor or competence that has to be maintained in order for certain structures to kind of keep that ability to regenerate over long periods of time. And we know in the axolotl that extends over their entire lifetime. They can regenerate throughout their lives.
Is there a part of the human body or an aspect of the human body that you think would be the easiest thing and maybe most necessary to be regenerated?
I think there is amazing progress being made every day. Just recently, there have been studies talking about regenerating human teeth. And that to me seems like something that would dramatically improve human health and the ability to kind of harness stem cells, to harness our own physiology in order to kind of spur new complex growth, I think is pretty amazing. I think at our level, even though we're thinking about regenerating limbs, a part of the process of regeneration is also just making sure that scarring doesn't occur. So if you do have scarring, if you have some kind of deposition of kind of scar tissue, that really does prevent regeneration from occurring. So even as a first principle step, the idea that we could decrease scar formation would already bring big gains to human health and well-being.
Doesn't scarring help to heal? I guess that you're saying if it's healing, then it's not regrowing?
So we know that for injury, there is that first stage where you are recruiting immune cells to make sure that you clear away any kind of pathogens or debris. You have to recover the open wound with the epithelial covering. And then cells will come in and they'll start to deposit kind of the transient or the intermediate kind of structure. So, cross linking of collagens to kind of create this matrix that helps kind of at least restore the function of the tissue. And so that would be something like a scab, or, if you had a kind of a cut on your skin, you would see kind of this doesn't quite look healed, but it doesn't quite look like an open wound anymore. And so then really the third step is to remove that transient structure and kind of restore the norm there. But what happens when we have scars is that kind of intermediate structure becomes built up. It becomes kind of stratified. So those the collagen that is kind of laid down during the healing process becomes really thick and becomes kind of stratified and cross-linked, and it doesn't get remodeled or taken away. And so just the presence of that kind of intense collagens, that intense kind of transient structure kind of occludes the remodeling, the restitution, the reconstitution that we would see during regeneration. And what we don't see in regenerating organisms is that kind of that heavily cross-linked collagen intermediate step that instead they kind of undergo that first step just like we do. They kind of recover the stump with the epidermis. They bring in immune cells. We know the immune system is really important for regeneration. But then they start to bring cells to the wound site that are going to basically start to reconstitution process. So there is really a divergence where when we start to lay down a lot of those heavy collagens, that intermediate structure, a lot of times that is kind of that diverts healing towards scar formation, and then it never gets remodeled into a state where we can actually restore some of the structures.
If someone didn't scar, would that mean that would be the starting point in order to then get to the point of regeneration, or does it have other benefits?
I think it has numerous benefits. I mean, there are a lot of long-term medical issues that folks deal with due to scarring. So there are kind of like keloid scars, and there are other types of hyperscarring, and scar formation can cause chronic pain over time. Of course, some aspects of that intermediate injury state is really important because, again, yeah, it does protect us somewhat. And there is a lot of ideas that potentially over the course of evolution, it's much better to survive with a scar rather than waiting to regenerate and potentially not surviving. But at this point in the process of medical history, it might be actually really beneficial to learn how to reduce scarring. And that could lay the foundation for either promoting our own regeneration or at least laying the groundwork for kind of interventions like stem cell therapies or gene therapies.
How did you get into this field?
I've been a biologist since I was growing my limbs to adulthood, and my training is really in cell and molecular biology. I was looking at cells in a dish for my PhD, and I really thought about like what are some of the extreme things that cells have to do together as a cohort in order to, make organisms function. And really regeneration seemed like one of those really exceptional things that cells really come together to make this process happen, and it's incredibly exceptional. That's really what sparked my curiosity about regeneration, and it led me to postdoctoral fellowship where I was actually studying these animals, these axolotls, and their ability to regrow limbs. And so that's where I learned how to use these animals to manipulate them and kind of couple my own expertise to apply to this system. And that's kind of what we've established my lab here at Wake Forest to do.
Thank you so much for taking the time to speak with me today. I really appreciate it. This topic is fascinating, and thank you for giving me a little bit of a window into understanding it more. And I look forward to seeing more of your research.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.